Good afternoon to everybody. Many thanks, Alain, to invited me to Amsterdam. I'm sorry, and I have to apologize because I'm both French and Italian, so my English will be not very good. So I will discuss about the clinical level. Even I'm not a physician, I'm a pharmacologist, and I work in Gustave Roussy Institut near Paris. And I will give you a kind of an update of the implementation of personalized medicine, or maybe precision medicine. Maybe we will have to focus on precision medicine in France with the experience of our institute in the field of oncology. As you may know, and maybe we have seen this this morning, standard anti-cancer therapy based on chemotherapy, radiotherapy here, and surgery reached a plateau in the end of the last century. In the last 10 years, the evolution of the omics with genomics, proteomics, and so on, metabolomics, became a reality even in clinical practice, at least in oncology. Among the 2.8 million cancer patients treated for cancer in EU, some have been will be cured with this standard therapy and with genomic application, but a large part of this patient will not be cured with this standard therapy and will need new genomic application to be treated or cured. In the last 5 years, with the example of the breast cancer here, high-throughput technology with CGH array, next generation sequencing, and allow from a patient biopsy to improve diagnosis, prognosis, to define new targets, and to better understand and better use the conventional treatment. The molecular circuitry of cancer cell is better understood despite its complexity. But nowadays, specific genetic traits can predict for the successful of a molecular targeting agent, such as you can see on this impressive, at least for a clinician, waterfall plots here with BRAF inhibitors in melanoma and with ALK inhibitors in non-small cell lung cancer. Nowadays, we are able to identify and then hit a target from a patient with a tumor specimen, constructing a molecular profiling in order to identify the molecular alteration, and then with a targeted therapy to give the right treatment to the patient. In France, with the input of INCa, the Institut National du Cancer, 9 ongoing precision medicine programs are ongoing. Some of them are called Saphir and Moscato, and we will see them after. They plan to include more than 3,000 patients in all tumor types. The concept of this molecular screening is developed drugs in a population defined by a biomarker identified here with a target identification from high-throughput technology for each patient. The first clinical trial, which was proposed by Professor Fabrice André, was Saphir 0.1 in the breast cancer with whole genome sequencing and Sanger sequencing here, in order to identify a targetable genomic alteration treated by a targeted therapy. In other solid tumors, the Professor Jean-Charles Solia of Gustave Roussy proposed the Moscato trial to patients included in phase 1 center with the implementation of next generation sequencing to find the best treatment for these patients, for each individualized patient. The molecular screening programs, more recently, allow enrichment of phase 1 and phase 2 trials in patients presenting genomic alteration. These programs are now benefiting to the patients, but molecular screening programs feed database and to develop algorithms for personalized medicine. This is a difference between stratified medicine and personalized medicine. From the first generation of molecular screening program, some trials aiming to evaluate bioinformatics algorithm to identify not only one target for the stratified medicine, but several targets for each patient in order to propose the best cocktail of drugs for treatment of their own cancer. These trials are now open in France and are delivered to our patients. They are called SAFIR 02 for breast cancer, with the targeting therapy according to more than 50 molecular alterations. This is proposed by Professor Fabrice André. And the second one is SAFIR 02 in lung cancer with Professor Jean-Charles Soria. And there is here several kinase inhibitor to target the right molecular alteration. So what are the economic issues? Are we able to finance this access to innovation in the field of oncology, and especially in France? We have to distinguish stratified medicine and personalized medicine. As you can see, stratified medicine develops a drug in a population defined by a biomarker. This is obvious. This stratified medicine can't, to our opinion, be financially modelized. However, with the personalized medicine, which aim to define a patient as unique and to not to taste a drug, but to test an algorithm, the personalized medicine is a revolution and a paradigm with impact on health policy, which is very difficult to modelize, to our knowledge. If the drugs were defined since the end of the last century by price alone, the genomic revolution drives treatment decision with, for the use of the drug, of their reimbursement with high price here in 2010, and with innovative drugs, but with limited public health budget. As we can see here, with the discovering of only two alterations with two drugs, gefitinib and crizotinib here, there is an increase, a significant increase of the budget for the treatment of lung cancer. How can it could be in the next future with more than two alterations for this treatment? With more than 1,000 new cases diagnosed each day, with about 20 targeted molecular therapy on market in France, and more than 400 currently under clinical phase development, the total cost of cancer in France was reached 15 billion euro in 2013, with 2.5 billion euro for the anti-cancer drugs. How can we promote a large and sustainable access to innovation in this context of molecular medicine and even personalized medicine? We, with my colleagues from ESSEC Business School, we work on this field, and we address this question on 6 points. New model of reimbursement with performance-based risk, sharing contract, reimbursement according real-life efficacy, and post-registration studies have to be built, even in France. Cost effectiveness and cost utility studies have to be submitted now by the firm to the French National Agency, ANSM or HAS, Haute Autorité de Santé, and then the price is given by the CEPS committee. And we have also with this particular medicine to improve the good medical practice with therapeutic standard use, called RTU in France, and also with therapeutic drug monitoring as pharmacologists. molecular platforms have been defined and elaborated by the ENCA. There are 28 in the territory of France, and they have conducted more than 150 tests in 2002. They have to now to improve their industrial process of these molecular pathology platforms, and there is a need to improve the process of the biological resource sampling instead in annotation, storage, and use. Another statement was the long process evaluation of the innovation companion test. We propose a fast-track procedure for these innovative drugs and their companion tests, and a better management of the biologic nomenclature, which is called NABM in France, to accelerate the reimbursement of innovative tests. We promote also, especially in our institute, the translational research for better patient care in order to develop clinical application and to favorite or to promote investigation on biological pathway in order to find new targets and new drugs. That was the case with the French government initiative, which have been construct this financing of the health policy with translational research here and field in innovation here conducted by ENCA with several tools and evaluation which are financed by the ENCA and the French ministry. They finance platforms, molecular platforms, they finance CLIP, which are early drug development center in the hospital. And we have in France with the ENCA the possibility to have the ATU system, which is temporary authorization system. And also the out of nomenclature test. All these structures and platforms have been financed by ENCA, and also with ENCA, the French government elaborates the third planned cancer last year. And for the Gustave Roussy Institute, we have construct and build molecular medicine building last year with the finance of the foundation, which are charity, and with private and public partnership in order to work here with the researcher and the clinician at the level of molecular medicine. Many thanks for your attention.